All right, in this part of chapter 12, what we're going to talk about is what happens to the cell cycle when things go poorly. Okay, so it's basically talking about how mitosis and cancer are related, in that when errors in mitosis occur, the cell starts to cancer and you start giving rise to cells that have mutations and so forth in them. So the essential idea, cell division is essential, of course, but we need to control it, and that was kind of like the last part that we were talking about, parts 1, 2, and 3, those types of things, the main idea. And this is what happens when it's not controlled, when we have an error and it gets propagated. Okay, so cancer cells themselves, these are obviously cells that have errors in them, and there's a lot of mutagens that give rise to cancer cells. And of course, mutagens are things that cause mutations, and chemicals, in the environment, cigarette smoke, things like that, are certain types of chemicals. UV radiation, okay, from the sun, can cause damage to DNA and, of course, skin cancer. Ionizing radiation like X-rays and gamma rays, things like that can really damage the DNA. And if the DNA is damaged beyond repair, that's a bad thing for a variety of reasons. But if some of that damage goes undetected and those cells start to kind of propagate themselves, that's where you can kind of get these errors occurring that can give rise to tumors and cancer. And the mutations themselves can also arise spontaneously just due to general errors that occur just from the sheer numbers of cells that we're actually dividing, and the sheer number of nucleotides in the genome that need to be replicated, etc. Okay, so cancer cells themselves don't respond to the body's normal kind of control mechanisms that were in place that we talked about in the first couple of sections here. Okay, they don't listen to these normal signals that regulate the cell cycle, those checkpoints, the different things that are occurring to ensure the integrity of the genome. These cancer cells aren't responding to those mechanisms. And some of the cancer cells themselves can make the things that they need really to kind of propagate themselves, to get themselves to grow and divide, and others kind of need growth factors that are made by the body itself just as a normal part of its metabolism, and these cancer cells can feed off of that as well. So some cells, for instance, some cancer cells respond to hormones that your body makes naturally, and that fuels their growth. Other cancer cells can just manufacture the things that they need to really sustain themselves. Okay, so cancer cells also fail to exhibit what's known as density-dependent inhibition. In other words, when these cells start to kind of run into each other, so to speak, they realize that there's enough of them, and that kind of shuts down the process of growth and division. And there's certain types of cells that really don't respond to this crowding, and they continue to grow and form these tumor cells, or tumors, really, that are these collections of cells that aren't responding to the normal mechanisms. Normal cells in your body typically divide 20 to 50 times in the process of growth, division, aging, and so forth, and then they die. And certain cancer cells are immortal in that they divide well beyond that 50 times and continue to divide, in some cases, forever, and give, if they're given a continuous supply of nutrients. And HeLa cells are a common example of this, and they came from a lady named Henrietta Lacks, and they were kind of taken from her unbeknownst to her. She had a tumor on her cervix, and when the doctor went in and was doing the biopsy, this was occurring during the 1950s. When he was doing the biopsy, he took the cells and then they cultured them in a dish, and what they found is that these cells continually grow and divide and really are not responding to any type of cell cycle control system. They just continue to grow and grow and grow if you continue to give them the nutrients. And one of the things with these particular cells, and it's just kind of an aside here, but you'll learn about it, and it's really kind of plays into the IB way here in the TOK type of a discussion and so forth, is that Henrietta Lacks was a poor person from inner city Baltimore, and she had this tumor, and these cells were taken from her, just for the biopsy purposes. But then when they found that they are really immortal and that they continue to divide, they've given rise to a whole field of biology and biotechnology and so forth. And you can buy HeLa cells today that are direct descendants from this lady. And the biotech industry has made billions of dollars off of these cells, while she and her family largely lived in squalor. And she died from cancer, but her family and her descendants of her family are still alive, and largely from the book that I read on this, The Immortal Life of Henrietta Lacks, her family isn't very well to do at all, and they live in some cases in poverty while people have profited off of these types of cells. So from the original diagnosis, sure, it was okay to take these things, excuse me, but is it okay to continue to use them, and is it okay to continue to use them without them knowing, without their consent, and to make billions and billions of dollars off these while the lady and her family don't profit off of it at all. So again, these cells can be used, and we'll talk about how they're used in biology in a much later topic, but these HeLa cells are really a topic of discussion. Okay. Now, as far as cancer cells themselves, and we'll go through this in class as well, but they have a very high mitotic index. In other words, they grow and divide at a much higher rate than a regular cell. And cancer cells are usually undergoing mitosis very rapidly compared to other cells. And then, for instance, like if you have a tumor cell in your brain or whatever, your brain is all neurons, and you learned in an earlier section here that nerve cells are in G0; they're not supposed to be dividing. And if the nerve cells are dividing, then of course they have a high mitotic index. And compared to the mitotic index of a normal nerve cell, it's very high because it's 0 in a normal cell, and then that would indicate the presence of a tumor. Okay. So primary tumors are the kind of site of origin of the particular tumor, and then as it begins to grow and spread, that's called metastasis. And we say that these tumors can metastasize or spread to other parts of the body and form these secondary tumors. Okay. During the process of cancer development, there's kind of a multi-step model in place that kind of explains how this occurs. And what you get are a lot of changes really occurring in the DNA, and this cell starts to become cancerous because what happens is the even though that damage has occurred in a cell, it's also perhaps affected other parts of the cell that really controls the spread of these errors and things that we've talked about earlier. But before we kind of talk about a lot of these things, what we have to discuss is some of these vocabulary terms that will help us kind of understand this. Okay, so when we're looking at these terms, we're going to talk about apoptosis, we'll talk about gatekeeper cells, caretaker cells, we'll talk about the P53 gene, we'll talk about just general oncogenes and proto-oncogenes, as well as tumor suppressor genes. Okay, so apoptosis, hopefully you know this by now, this is the programmed cell death. This is the normal process that occurs during cell growth and division, just to ensure that these faulty cells don't divide. Any error that occurs, apoptosis should ensue and prevent the spread of this faulty organism. So this is a normal process within living organisms. Now, there's two general kind of groups of tumor suppressor genes. You have gatekeepers and you have caretakers. Okay, with gatekeepers, what these do is they act as a set of brakes on this cell proliferation. In other words, these cells grow and divide, and these gatekeepers can kind of act as a set of brakes to ensure that these particular types of errors, should they have occurred, don't get spread into daughter cells during the process of division. And then these caretaker genes, these are genes who have products that really protect the integrity of the genome. In other words, they're going to repair, they're going to care for the DNA within that cell, and their job really is to repair this damaged DNA. And if a gatekeeper gene gets mutated, damaged somehow, whatever, what they do is they lose their ability to kind of get these repair mechanisms going. So you can see that if the gatekeeper genes get mutated, and a mutation occurs in a different spot in the genome, there's no way to kind of care for that, so to speak. So what happens is those mutations can start getting propagated, and if they don't go, I guess, through the normal process of growth and repair, this is what's going to give rise to a tumor. Okay. So what we were talking about, these mutant caretaker genes and their ability to repair for the or to care for the genome and repair the damages that occur. We've probably all heard of the breast cancer genes, and they're BRCA1 and BRCA2. And what happens is if you have a mutation in those, you lose the ability to kind of care for the genome, you lose the ability to stop the cell and repair the DNA or get rid of the cell through apoptosis. And as a result of that, people who have errors in BRCA1, BRCA2, or both are at a much increased likelihood of developing breast cancer or ovarian cancer or both. So they have to go through numerous checkups, and there's lots of ways in which you can go about, you know, I guess ensuring that you don't have breast cancer, you don't get ovarian cancer, those types of things. And those are all, you know, subjects of, you know, conversation, I guess, with family members, people who have these things, medical professionals, etc. But that's just an example of these gatekeeper types of genes, these caretaker types of genes, and how they're involved in things that we know about, you know, from our daily experiences. All right. So now P53, this is a tumor suppressor gene, and this is like a big one. This is known as the guardian of the genome because it acts as a gatekeeper, and it really protects the vast majority of the DNA really within the body, because it makes this protein called P53 protein that can do a number of different things. Chiefly what it can do is it can stop cell growth and division so that we can hold the cell at a particular phase of the cell cycle so that we can check this damaged DNA, repair it, or get rid of the cell. So it does a lot of different things in terms of kind of suppressing the formation, growth, and spread of a tumor, okay? And when this P53 protein again binds to the DNA, it's going to regulate the expression of the genes that control the cell cycle. And we're going to kind of go through a paper that kind of shows how cigarette smoke and smoking in general causes cancer. And P53 will kind of be at the root of this discussion here. Because what it can do is it can activate these repair proteins when DNA damage has been sensed. And if you can't repair that DNA through some of those other mechanisms that we've talked about, this P53 protein has the ability to kind of kickstart apoptosis and get rid of that cell before it becomes, you know, cancerous and a tumor and that sort of a thing, okay? But once P53 itself becomes activated, it can also, in addition to some of the things we just described, it can also activate a number of other different genes within the genome that help do kind of many of the same types of things that it does. And one of the notable genes here is P21, and it's going to kind of, those are going to be some repair types of proteins that are going to do a lot of the same types of things that P53 does as well, okay? And these tumor suppressor genes, what they're going to do is you know, prevent the propagation of these mutations. But if they themselves become mutated, you can see how they're going to then lose their ability to do all of these important jobs, and therefore these errors that occur are going to get propagated. We're going to have the formation of tumor, and we're going to have all sorts of bad things that occur, okay? Now with oncogenes and proto-oncogenes, proto-oncogenes are the name given to the normal kind of form of the gene. And this is a normal gene that if it becomes damaged is going to, you know, give rise to tumors. In other words, these proto-oncogenes are normal genes that promote, regulate, and kind of, I guess, look over normal growth, development, cell proliferation, etc. And then when they become mutated, when a proto-oncogene becomes mutated, it forms an oncogene. And this is what it can lead to the cancer, because now these normal genes that are controlling the cell cycle have become, you know, mutated. They've become oncogenes, and now the cell cycle just kind of goes down this runaway path because there's no regulation occurring there, okay? And if it doesn't get repaired, as we said, those proto-oncogenes become oncogenes. And under normal conditions, what happens is these cells should go through this programmed cell death when they're damaged beyond repair. But if this proto-oncogene has become an oncogene and they've become activated, now you're just promoting this cell proliferation. You've got this increased expression of the genes, and it's very easy to see how this damaged DNA really kind of leads to this runaway cell growth and in the formation of a tumor, okay? These tumor suppressor genes now, these are found within the genome, and again, they're going to inhibit or suppress the formation of a tumor. And they're going to encode certain types of proteins that bind to this cell cycle machinery and prevent this runaway cell division. And if these tumor suppressor genes become mutated, then you can see how they don't do the things that they're supposed to do, and that's what's going to give rise to formation of tumor and all of the other bad things, okay? So we'll pick up here with part 2 and kind of explain how this all fits in with mitosis. So, you know, there was a lot there, a lot of things, a lot of explanation perhaps that we need to go through in class, but it'll give you a basis of things to kind of start with, and then we'll kind of broaden our discussion in class. So if you have questions, of course, write them down and be prepared to talk about it. Okay? We'll see you soon.